Hello everybody, my name's Nate from the Rowett Institute in the University of Aberdeen, and today I'm going to talk to you about our wonderful gut microbes and to introduce the exciting concepts that underpin my PhD project. You can see me here happily admiring my bacteria, trillions of which live on and in us and play a huge role in our health and also in preventing infections. Our bodies are colonized by numerous microorganisms, collectively known as the microbiota. The types of microbes vary between different sites in our body, but each site harbors millions of microbes. Today, my focus will be on our gut microbiota and how it specifically contributes to our health and to preventing infections. Now, with all of these microbial cells, you might ask, who are we? The statement that we're 10% human and the rest of us is comprised of microbial cells is a myth. There are approximately 38 trillion cells in the average human body and approximately the same amount of microbes. And I suppose you could say that makes us 50% human instead. Even with this 1-to-1 ratio, our bodies are getting pretty crowded. But who's in that crowd? It's mostly bacteria, and they make up 90% of the microorganisms that inhabit us, and mostly belong to the Firmicutes and Bacteroidetes phyla of bacteria, but there are also microscopic fungi and viruses too. I will mostly discuss our gut bacteria, and I will briefly mention our gut fungi, but you should check out Anne Kristen's talk for more about fungi instead. It's important to remember that not all bacteria are bad. Bacteria, as with other microbes, exist on a spectrum, with some causing infections. Others are extremely useful and essential for our health. Some are opportunists and may cause infections if certain factors are involved, for example, if they end up in the wrong body site or suddenly expand in number, and I will discuss why this might happen later. So how do we get our microbiota? In the uterus, we are sterile and begin to develop our microbiota as we are born. Babies of vaginal birth will have a microbiota that reflects their mother's vaginal and fecal microbiota, whereas C-section babies will be predominantly colonized by their mother's skin microbes. This microbiota is relatively stable in adulthood, but is still susceptible to change by a number of things. One of the largest influencers of microbial composition in the gut is the nutrients that we intake. The nutrients that escape digestion by our bodies and reach the gut are what our microbiota feeds on, and just like us, they have their preferences. For example, high fiber diets will encourage the growth of Firmicutes, while high fat diets will enhance the Bacteroides genus. And diet can be influenced by geography and lifestyle choices, which is why we also see geographical and lifestyle variance in the composition of the microbiota. For example, generally, in Western diets, they are higher in fat and lower in fiber, and so we see an increased abundance of Bacteroides and bile-tolerant species of bacteria. Another influencer is antibiotics, which are quite often broad spectrum, meaning that not only will they target the bacteria that is making you ill, but will also target your friendly gut bacteria. And it's the loss of these gut bacteria that means that we also lose all of the benefits they have on us. What are these benefits? What makes the microbiota so important? Well, some of the foods we eat are undigestible to us. Often lumped into the term fiber, these complex carbohydrates are digested instead by our resident bacteria. Without the microbiota and its ability to digest our leftovers, we would be lacking some pretty important biomolecules that are essential for health, such as vitamin K, B group vitamins, but also neurotransmitters like dopamine and serotonin, which regulate our moods. And products of microbial fermentation, in fact, can also stimulate hormone production and play a role in the hormonal balance of our bodies. However, the most important and major byproduct of bacterial fermentation on our leftovers are short-chain fatty acids, or SCFAs, predominantly acetate, butyrate, and propanoid. 90% of these SCFAs are absorbed by the human body and have important physiological effects. Acetate is readily converted to more butyrate by the microbiota, and it's a good thing too, as butyrate is a major energy source of the cells that line our intestine. And SCFAs can also stimulate the immune system and prime it to be ready to act when needed. In fact, our microbiota and immune systems are intimately linked. During immune development as babies, our developing and fluctuating microbiota effectively trains the immune system to be able to appropriately respond to invaders. So you see, when our bacterial communities get disrupted, we lose so many of the benefits afforded to us by this symbiotic relationship with our microbiota. However, the most interesting benefit of the microbiota from my point of view and from the perspective of my PhD project is its ability to prevent infections. This ability to prevent infections is known as colonization resistance. The term was coined in 1971 when a series of observations showed that mice were more susceptible to infection by salmonella after antibiotic treatment, and this effect was linked to the depletion of their microbiota. There are several mechanisms in which the microbiota protects us, and I'm going to discuss that in the next few slides. First off, the sheer numbers of microbes present in our guts creates a physical boundary against potential pathogens. They block off any sites on the body surface that pathogens could latch onto and thus invade our tissue and cause infections. In addition, all of these microbes mean that there is some serious competition for nutrients. The importance of this nutrient competition is highlighted by studies that have shown that when disruptions cause reduction in the numbers of firmicutes and bacteroidetes, you see an increase in succinate, another SCFA, which is usually readily taken up by the microbiota. This increased succinate then acts as a nutrient source for opportunists like Clostridium difficile, which can overgrow and cause infection. So changes in the bacterial communities also results in a change in the landscape of secreted compounds, and that might include short-chain fatty acids, antimicrobial peptides, or immune regulators. And these can either directly inhibit the growth of pathogens or prepare the body's defenses against them. All of these mechanisms that we have discussed can also apply to fungal infections, but little is known about the relationship between host bacterial communities and the fungal component of the microbiota. And so this is the challenge of the FunHoMic group, to try and decipher this relationship using the pathogenic yeast Candida albicans as a model organism for our studies. The gut is considered to be a significant reservoir for Candida albicans, which can live harmlessly in our guts, but is one of these opportunists that we discussed. In my project, I hope to be able to understand how specific species of bacteria contribute to colonization resistance against this yeast, and to pave the way for developing therapeutics to prevent and treat Candida albicans infections. And thank you for listening.